And now we have Chapter 8, Microbial Genetics. But this type of genetics is not what we covered the last time. It's not that simple, DNA to RNA to protein, and we'll see why in a sec. So, what is vertical gene transfer? What is horizontal gene transfer? And when you read the sentence, it doesn't really illuminate too much. It says vertical gene transfer occurs during reproduction between generations of cell. Okay, so when you have vertical, it's up and down. And what they're trying to say is if you have a bacteria that doubles its size and does binary fission, and it gives rise to two new daughter cells that get big and fat and divide and do binary fission, and now you've got 4 bacterial cells, and those 4 bacterial cells get big and fat and they divide in half, and now you have 8 bacterial cells. That's vertical gene transfer. Meaning that the original cell had one circular piece of DNA. It made a copy of it, so then when it did binary fission, you got 2 cells, and they both have their own one circular piece of DNA. All right, in fact, that's what usually happens in mitosis in our body, and it also happens when we make egg and sperm and do sexual reproduction in our body. But bacteria can do something different. And the difference has a lot to do with the fact that they are very simple. They are the first organisms that arose in the primordial soup 4 billion years ago. They're still around, highly successful, but they don't do sexual reproduction. So the reproduction is basically cloning themselves. And if that's all they did, they probably would die, because you can't keep a population very robust if they start to slide down where you're just cloning yourself over and over and over and over and over again. There's no change, there's no diversity, and then something changes in the environment, and then bam, they're dead. So, okay, if you can't do sex and you can't die, then how do you get different? And the way that bacteria get different is horizontal gene transfer. Now, people will argue, yeah, with vertical gene transfer, you can have the occasional mutation, but that isn't really going to give you large changes all the time. If anything, bacteria are able to thrive under nuclear war. They're fine with fire. They're fine with, I don't know, volcanic eruptions. They're fine with tornadoes and hurricanes. It's very, very difficult to kill all bacteria, and that isn't because of vertical gene transfer. The reason why you can't kill bacteria and you'll always get survivors is because of horizontal gene transfer. So let me just read it and then I'll explain. Transfer genes between cells of the same generation. In other words, neighbors. They're next to each other. This is also called gene trading, which means that bacteria can trade parts of their DNA with each other, something we cannot do. I like you very much, but I can't saddle up next to you and say, hey, here's a tip of my chromosome 13. Have a piece. But bacteria can, and this keeps the bacteria more robust. Okay, so what it really would mean is that with 2 bacteria that are neighbors that can touch one another, that they do have the ability under certain circumstances, and they always will create these circumstances, and the circumstances are stress and change. So bacteria get stressed and have to respond to change just the way we do, but they do it quite differently. They do gene trading, and we do sexual reproduction. And the other thing about gene trading that's very different, besides the fact that you can have 2 bacteria next to each other, and they can decide that on some level that they are going to give each other parts of their chromosome, during that period of time. They're not doing it during binary fission. They're doing it during a time maybe right after binary fission, but before you do the next binary fission. They don't have to be related. They can be a different genus and species. You could have an E. coli next to a Proteus vulgaris. I mean, that's unheard of in the animal kingdom. You have to mate with the same genus and species. Homo sapiens mate with Homo sapiens. Homo sapiens, don't saddle up to another creature in the animal kingdom and be able to have sexual reproduction that's going to be successful. But bacteria, they're in their own kingdom, and they have all kinds of genuses and species and all kinds of different categories. But that's us. We do that to them. We categorize them. But they don't. They just want to have some bacteria survive. And they're very altruistic about that. So that's the reason why gene trading, you could have an Escherichia coli saddle and be next to a Proteus vulgaris. And there's nothing to stop them from trading genes. There's nothing to stop them from what is called recombination. They're going to recombine the genes. And they're going to do it during their lifetime. And it only takes 2 minutes. And this is something that they do all the time. That's why we're never going to be one step ahead of them. Because as soon as things get a little bit on the edgy side, they're going to do gene trading like nobody's business because they want to have someone survive. It's not like E. coli goes, I only want to have E. coli survive, so I'm just going to settle up to other E. coli. It doesn't happen. E. coli bumps into bacillus, and guess what? If things are changing and they're under stress, then gene trading, here we go. Because their aim is to keep a member of their kingdom alive. And that is something that once you do sexual reproduction, you're kind of in a corner. You only have one shot at it at a time. And you certainly can't change your mind and decide that when you're 30, you want to recombine your genes. This isn't going to happen. But bacteria, it does, it can, it will. They do it all the time. So let's go into some of the gene trading, horizontal gene trading. It's horizontal because obviously they're next to each other horizontally, sort of. Anyways, off to the next slide. Okay, now this is a slide I'm not going to hold you responsible for. This is trying to show you how they came about figuring out that bacteria do this. Bacteria have always done this. It's just that we reach a point where we can figure out that they're doing it. And so in this particular experiment that I'm not going to hold you responsible for, what you have is you have this poor little mouse, and they are going to give them... Now, encapsulated bacteria tend to be very pathogenic. So they inject the poor little mouse with very pathogenic encapsulated bacteria, and the mouse dies. Then they're going to take a mouse and inject them with non-encapsulated bacteria, and they tend to be non-pathogenic, so mousey is fine. Then they're going to heat kill the encapsulated bacteria. And I will refer back to what actually happens because it doesn't seem like it's too logical. Now, when you heat kill the encapsulated bacteria, meaning the bacteria die. And that's the reason why the mouse is still healthy. And underneath, they show you the plates that, they're going to sacrifice a little mouse anyways. And then they're going to look to see if there are any bacteria. And if you notice with this little guy, there's no bacteria. But then they're going to take a mouse and they're going to take so they're going to take living, non-encapsulated bacteria and heat-killed encapsulated. And what you would expect is you would expect the mouse to be fine, just like the mouse over here is fine. But the mouse dies. And you recover live encapsulated bacteria. So how did that happen? And it happened through a process called transformation. I love the word, because to transform somebody means that they change, and usually transformation is a very positive thing. And for the bacteria, it is. So let me explain. I'll explain the definition, and then I'll go back to mousy land. So in transformation, a neighboring bacteria dies, degrades, and a small piece of DNA can be transferred to an adjacent bacteria, where it can, that little tiny piece of DNA from the donor that's dead, recombines with the recipient chromosome and thus changes or transforms the recipient cell. Um, it, it, it's just kind of fascinating how bacteria have evolved into this type of thing. So you're going to say, well, where do you get these populations? Well, look into your large intestine, look into your gut. This type of recombination occurs all the time because the amount of bacteria in your large intestine, in your colon, is amazingly high, amazingly high. Trillions upon billions upon quadrillions. So you're always going to have, just as a matter of course, some of the bacteria are going to die. The recipient living bacteria will inevitably have a little piece of DNA that will, from the dead donor, get inside. It'll combine with the main chromosome. Now, it kind of depends on what that information is. That information could be neutral and it could be beneficial, and it may not be evident that it's beneficial, but then all of a sudden things change and it's incredibly beneficial, and then the other bacteria die, but that one lives. So what happened with the mouse experiment is that when they combined the non-pathogenic with the pathogenic that they heat killed, um, they set up transformation. They didn't have any idea what they were doing, but when they heat killed the pathogen died, and in the test tube before they injected it into the mouse, all of these little pieces of DNA from the pathogenic bacteria were present. So when they added the non-pathogenic, non-encapsulated bacteria, transformation occurred. Little pieces of the DNA from the heat-killed pathogen entered the non-pathogen, and some of them became pathogenic. And that's the reason why the mouse died. So in transformation, it actually takes some, a phenomenon like dying, and yet part of the bacterial DNA will live on in the population that's still alive. So bacteria in your body, normal flora, are, some of them are gonna die, some of them are gonna live, but all of them that live will be transformed. And whether it's good, bad, or indifferent doesn't really matter. Again, it's very altruistic. What will happen is if things change and something catastrophic happens, then you're going to have the survivors be those that have some of the information from the dead donor as well as themselves. And that then gives them the leading edge, and they will do it all the time because they know if anything is going to happen, it's going to be change. And they do it to hedge their bets. And that way, someone's going to be a survivor. They don't care. And that survivor will grow and thrive and meet other bacteria and do recombination. So it's absolutely fantastic. We cannot do it anytime you have any organism that can do sexual reproduction. It's only when they do sexual reproduction, make eggs, make sperm, that this type of mixing of the genes, because you're half mom and you're half dad, but you're a unique individual. You're not your mom and not your dad. But you're a unique blend of them. So you are an experiment in recombination, but in sexual reproduction, that's the only time you can do it. But with bacteria, they can do it all the time. Conjugation is another way that bacteria can exchange genes with each other without their having to die. So to conjugate means to bring together. And here, and again, usually what drives conjugation is stress and change. So things are stressful, and you have one bacteria, because of stress, end up looping out a small piece, a small circle from their main circular chromosome. And It's called a plasmid, a plasmid. And it's given many, many other names, but I'm just sticking with plasmid. And what is on this plasmid, the genes that are on the plasmid, are antibiotic resistant genes. I'll tell you how incredibly smart that is to do at the very end. So you have the genes that encode for antibiotic resistance in here. And when this bacteria meets up with another bacteria that does not have a plasmid, it will form a bridge between the two of them. And once the bridge is formed, you'll make a copy of just the plasmid, and it will enter into the recipient cell, and then the recipient cell is now also going to be antibiotic resistant. And then they break the bridge and they go off their merry way. 2 minutes. So all it's going to take is 2 minutes. And it means now that you had one cell that was antibiotic resistant, and now you have two cells. And again, they can be totally unrelated. They don't really care. So this cell is still going to survive. That cell survives. They are also both antibiotic resistant, and then they can go on and do the same thing to other cells that don't yet have a plasmid. And that is an incredible way of responding to, let's say, a person taking an antibiotic and you're trying to kill a certain pathogen. And if the pathogen comes up against a non-pathogenic bacteria that has a plasmid, it takes 2 minutes to convert the pathogen into an antibiotic-resistant pathogen. This is again why we are never going to have that magic bullet for all bacteria. Again, they do this all the time. They do recombination, they do conjugation, and they will do this during their lifetime with their neighbors. Okay, so the next slide is goes into the terminology that I like, that will obviously be on the quiz. So in conjugation, the donor cell with a plasmid, this little circular piece of DNA that breaks off, that contains all the antibiotic genes, can bind to a neighboring recipient cell, which doesn't have a plasmid, and forms a bridge. Then the donor plasmid DNA makes a copy, replicates, and one strand enters the recipient, and that means that that cell now has all the genetic information contained on the plasmid and are now antibiotic resistant. So again, it's a very wonderful altruistic way, and it's very smart on the part of the donor bacteria. And of course, perhaps we understand more about this process and are looking at those bacteria that become antibiotic resistant because it's such a problem. There could be other genes, but we're not really looking at them. But when we do look for the antibiotic genes, when it becomes stressful, it's extremely smart on the part of the bacteria to put those antibiotic genes into a plasmid. And not keep them in a whole chromosome, because that way they don't lose it, but they also spread the joy, and that way all of the recipients of the plasmid are now endowed with resistance, which means that bacteria win the war. And again, they don't care who wins the war as long as they still have survivors. Okay, transduction. I know we haven't gotten to viruses yet, but bacteria are attacked by viruses as well. They have their own viruses, and we'll learn more about that in the next PowerPoint. But what viruses want to do is they want to replicate, and they have to hijack a cell in order to do that. And what bacteria do, since they only have one chromosome, it's very easy for the viruses to destroy that one chromosome, break it up, and make it into these non-functional pieces so that the viral DNA can take over the cell and make lots of copies of itself. And once it's used up all the, I don't know, energy of the cell, it kills it, breaks it open, and releases hundreds and hundreds of new viruses. Now, when that happens, sometimes there's a mistake. And the mistake is rare, but the mistake is almost always made. So what should happen is the little virus right here, and it has little pink DNA, and so it's injected into the cell and rapidly is going to destroy the host DNA. And then when it comes to package so when you package the new DNA inside the new viruses, of course you want to have it to be viral DNA, right? But sometimes they make a whoops and they package a little bit of the bacterial DNA. So when that little viral particle gets to a new bacteria, what it's injecting is bacterial DNA, not viral DNA. And that's the whoops. But now that little bacterial piece of DNA can enter the chromosome and change it. So even with the act of being under attack by a virus, with transduction, trans means across. So you have a bacteria, and then it's affected by a virus, and some of the bacterial DNA gets inside one of the viruses. That's the trans, and then that virus is going to try to infect another cell. Of course, it doesn't know that it has bacterial DNA rather than viral DNA, but it has bacterial DNA. So it's not going to cause a lytic cycle. It's not going to take over the cell, but the DNA that's been injected in by this weird little virus is then going to incorporate into the host chromosome and change the bacteria for, good, bad, or indifferent, but the change will be made. So now you have 3 ways of changing things. Let's go, yes, and you can read all of that. Blah, 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 blah. Okay, so in transduction, a virus enters a donor, causes a lytic infection, by accident packages a piece of donor DNA in one of their new viral particles. When that virus infects a new bacterial cell, it only injects the bacterial DNA, which can now incorporate into the recipient chromosome, which causes a change. So now what we have through this ability of bacteria to do these horizontal gene transfer feats is you have your transformation, you have your transduction, you have conjugation. And between the 3 of them, they are able, as populations, to grow, change, divide, adapt, figure out how to survive, be robust with all of these changes that are constantly going on around them. And it's made these organisms extremely successful. So even though they're very small and they're never going to get very big, they are remarkable. So if there was a terrible catastrophe like a meteor hitting Earth and everything under 1 pound dies, you're still going to have bacteria. So that's about the plasmids. So the plasmids, we're going back to the plasmids. So the plasmids are these little circular pieces of DNA. Stress tends to make them pop out, and they are going to make awfully sure that they have all of their antibiotic resistant genes on that plasmid. And that's what they look like. This is actually a electron micrograph in which they colored it because electrons are black and white, but they're very, very cool. And there they are. And this is just showing you way too much information as far as what the genes are. But, here you have TET, and that's going to be tetracycline resistance, and so forth and so on. So those are the plasmids. And that's it. So when I see you next, we're going to be talking about viruses. See you soon.